Hi friends, as you all are aware, we have started a new series on our channel, and that is graph related discussion. Today's video is on another graph, and that is myoglobin dissociation curve. But before we go to the graph, let us understand something about myoglobin. And I have given the title Myoglobin, the Unsung Hero. Let me explain why. Imagine that you are performing a continuous aerobic activity, continuous isotonic muscle contractions, slow but sustained type of activity. What happens in this activity? Alternate rhythmic contractions and relaxations of the various muscle groups, running, jogging, cycling, swimming, etc. Now in this case, if you have to sustain this activity for a long period, you need energy production inside the muscles for prolonged durations. So, which characters do we have in this entire scenario? We have hemoglobin, which takes up the oxygen from the atmospheric air, and via bloodstream it passes on this oxygen to the muscle cells. Then inside the muscle cell there is mitochondria. It will take up that oxygen and will generate the ATPs. But there is a catch here. As the muscles are contracting alternately, there is going to be a possible ischemia. The blood vessels inside the muscle will be compressed, and therefore the blood flow to the muscle is not going to be continuous. That means oxygen delivery to the muscle cells also won't be on a continuous basis. It means we need someone to hold the oxygen inside the muscle cell and deliver it into the mitochondria of that muscle cell on a sustained basis. And that is where myoglobin comes into the picture. Therefore, I call it unsung hero. We talk about the hemoglobin, we talk about the oxygen, we talk about the mitochondria, blood flow, etc. But myoglobin plays a very critical role whenever we have to perform a sustained activity, muscular activity. Let's look at the myoglobin first. It is the counterpart of hemoglobin in the muscle. That means there is hemoglobin inside the blood in the RBCs. Its counterpart in the muscle cells is myoglobin. In fact, it is also called as myohemoglobin. Myo means muscle. So it's muscles hemoglobin. That is how we should look at it. Particularly abundant in the slow oxidative muscle types. You know there are two types of muscles, the slow oxidative and fast glycolytic. When you want a quick burst of muscular activity, you will use fast glycolytic fibers. But when you want a slow and sustained type of muscular activity, these muscles are put into action. They are called slow because they are not causing very rapid contractions, and oxidative because they generate ATPs by oxidative phosphorylation, that is inside the mitochondria. So for prolonged low intensity activity, these types of muscle fibers are used. Okay, now myoglobin is an oxygen binding protein, just like hemoglobin. Even myoglobin takes up oxygen. It is heme containing protein. Yes, myoglobin also has heme, very similar to hemoglobin, apart from a few exceptions. It functions as a monomer. Now that is where it differs from hemoglobin. Hemoglobin, one hemoglobin molecule can take up 4 oxygen molecules. It has got 2 alpha and 2 beta chains. So 4 chains in all, and therefore it's a tetramer, and it can take up 4 oxygen molecules. Myoglobin is a monomer and homologous to either an alpha chain or a beta chain of the hemoglobin. Right, and therefore myoglobin binds only one oxygen molecule. One molecule of myoglobin takes up only one oxygen molecule. Now note this point, that the myoglobin has much greater oxygen affinity compared to hemoglobin. So hemoglobin will deliver its oxygen into the muscle, it will be taken up by the myoglobin inside the muscle cells, and it has got a greater affinity. It will hold its oxygen for whenever up to whatever time it's required to hold. In the muscle capillaries, hemoglobin hands over its oxygen to the myoglobin. So basically there is a blood vessel, there are RBCs, if you can see here, yes, hemoglobin inside the RBCs. This hemoglobin will deliver its oxygen into the muscle cell. There is a muscle cell shown here, representative diagram. So oxygen delivered, myoglobin will take up this oxygen, and myoglobin will pass on this oxygen to another myoglobin molecule, and so on. And finally, the oxygen will be delivered to the mitochondria, where ATPs are being generated. So this is how oxygen transfer occurs. Transfer from hemoglobin to myoglobin, and then so on. Now this speeds up oxygen diffusion through the muscle cell. You know, oxygen by itself has got low solubility. In other cells, it's okay that hemoglobin straight away delivers its oxygen inside the cytoplasm, that is inside the cell of any other cell, and then it will eventually enter the mitochondria and ATPs will be generated. But muscle needs to generate ATPs rapidly and on a sustained basis. And the problem with this oxygen is that it has got a low solubility. So what happens is, even if hemoglobin is delivered into the muscle cell, because of its low solubility, the PO2 in the muscle cell cannot rise that rapidly. Point number one. Point number two is that we need to create a great concentration gradient for oxygen inside the muscle, so that from the muscle, from the cytoplasm, the oxygen can be delivered into the mitochondria because of a large concentration gradient. And that large concentration gradient, please note this point once again, from the cytoplasm to the into the mitochondria. If there is a large concentration gradient for oxygen, then oxygen delivery to the mitochondria can be very rapid. And that is where the role of myoglobin comes into action. It holds the oxygen and it creates that large concentration gradient for oxygen from the cytoplasm into the mitochondria. So oxygen transfer into the mitochondria will be that much faster. That's the importance of myoglobin. It speeds up the diffusion of oxygen through the muscle cell, through the cytoplasm of the muscle cell. Now oxygen has low solubility, as we have already mentioned. There is insufficient dissolved O2 by itself to establish an intracellular O2 gradient. Look, because oxygen has got low solubility, even in the cytoplasm of the muscle cell, it will dissolve slowly, and therefore it won't be able to create that gradient of oxygen to be delivered into the mitochondria. And therefore there is no large enough gradient, as we have said already. Myoglobin acts as a temporary store of oxygen in the muscles. It's a temporary store of oxygen. So oxygen is stored, and whenever muscular exercise happens, alternate contractions and relaxations. So with contraction, there is a possible ischemia because of the blood vessel blood vessel is compressed. When the muscle contracts, the blood vessel inside that muscle will be compressed, and in this ischemic condition and with low PO2, because blood flow is intermittent, there is period of ischemia. On the other hand, muscle has already consumed oxygen, it is performing a continuous activity, and therefore this is a condition of low PO2 in the muscle. In such a scenario, myoglobin will release its oxygen so that muscle or mitochondria inside the muscle get a constant supply of oxygen. constant supply of oxygen in the mitochondria of that muscle cell. That's the crucial role played by myoglobin. All right. So during muscular activity, as the muscle contracts, I think it's a repetition anyways, ischemia, muscle can use oxygen supplied by its myoglobin. So even if there is less delivery of oxygen because of that intermittent contraction of the muscle and compression of the blood vessels, muscle still has a good enough store of oxygen, and that is thanks to this unsung hero, the myoglobin. So I repeat my sentence again, myoglobin is an unsung hero. We won't be able to perform slow and sustained activities, we won't be able to run the marathon runs if there was no myoglobin. All right. Myoglobin releases its oxygen only under conditions of very low PO2. Yes, another important factor for myoglobin is that if the PO2 is very low, like in the condition of muscle ischemia, as we are contracting the muscle, PO2 is going to fall. Muscle has already consumed the oxygen. Oxygen is less inside the muscle, or rather less, we would say it is not getting supplied continuously, and therefore a condition of low PO2 inside the muscle cell, and that's when myoglobin can release its oxygen. That is the property of myoglobin. And now we come to the main part of the discussion, and that is myoglobin dissociation curve. Actually, this video is on the graph myoglobin dissociation curve. So first things first, you have read about the oxygen dissociation curve, which is S-shaped or sigmoid shaped. Myoglobin dissociation curve is a rectangular hyperbola. You can see here the curve shown in the blue color is the myoglobin dissociation curve, and compare it with hemoglobin, oxyhemoglobin dissociation curve, which is a S-shaped. So why is it that it is a rectangular hyperbola? Let's understand the graph. Look, one hemoglobin molecule takes up 4 oxygen molecules, right? So imagine that there are hemoglobin molecules coming into the lungs. Right now there is no oxygen taken up. So each hemoglobin molecule starts taking up oxygen. Okay. First oxygen molecules combined means 25% saturation. Second oxygen molecules combined with hemoglobin, 50% saturation. And then third is 75%, and fourth oxygen molecule, 100% saturation. So that is what is the saturation of hemoglobin. What about myoglobin? Myoglobin, one molecule can take up only 1 oxygen molecule, which means what? Check it out here. On the vertical axis we have percent saturation. So when 1 oxygen molecule binds with the myoglobin, 100% saturation, and therefore you can see that this part of the curve is rising almost instantly and reaching almost instantly to the peak, because as soon as 1 oxygen molecule combined, it means 100% saturation, and therefore the steeply rising part of the curve tells us that oxygen molecule, 1 oxygen molecule each, has been taken up by the myoglobin molecules, and then 100% saturation. So that's maintained there. This is the reason why it's a rectangular hyperbola. So if you are asked in the vivas why this graph is a rectangular hyperbola, the answer is that only 1 oxygen molecule binds with the myoglobin. So when it binds, it's like 100%, and therefore the graph rises instantly to 100%. 1 oxygen binding with myoglobin. Right. Also, now this is about the oxygen binding with myoglobin, oxygen association with myoglobin. What about oxygen dissociation from myoglobin? How the oxygen dissociates or is released by myoglobin? On the horizontal axis you can see it's the PO2. So PO2 100, 90, 80, 70, 60, 50, 40, 30. PO2 is falling from 100 to 30 or 20. The graph is not coming down at all. That means at a high PO2, myoglobin holds its oxygen. So when hemoglobin is rapidly delivering its oxygen and the PO2 is rising, myoglobin is holding its oxygen inside the muscle cell. And then when there is a low PO2, you can see that PO2 20, 10, 5, the graph is coming down very steeply. So this time we are talking about from the other end of the graph, how the graph is coming down steeply when the PO2 going below 20. It means in the condition of low PO2, myoglobin liberates its oxygen, and that is what is required, because muscle has a intermittent blood flow during the continuous activity, and therefore there is a possibility of low PO2, and therefore a sustained O2 provision to the mitochondria is the job of myoglobin. So you can see here below PO2 of 20 up to 20, the graph is almost flat, it's not coming down, which means what? Saturation of myoglobin is not coming down. But PO2 20, below 20, and see the graph is coming down steeply. That means below the PO2 of 20, myoglobin is quickly releasing its O2. And as it's releasing, myoglobin molecules are releasing the oxygen molecules, the percent saturation steeply comes down. Where is it releasing? It is releasing the O2 into the mitochondria of that muscle cell, so that the ATP generation can be sustained. So that is why I said myoglobin is an unsung hero. Okay, so that's about the myoglobin dissociation curve. Just as a quick recap, why it is rectangular hyperbola. First, the percent saturation rises very rapidly to 100% because only one oxygen is to be taken up by the myoglobin molecule. So binding of one oxygen molecule, 100% saturation. That's the reason why it is rectangular hyperbola. Saturation maintained after O2 has combined with the myoglobin. And then we saw why, how oxygen dissociates from myoglobin. High PO2, that means O2 was delivered by the hemoglobin into the muscle cells, therefore there was a high PO2. Myoglobin takes up and holds its oxygen. And when there is very low PO2 in the muscle, period of ischemia, myoglobin releases its oxygen into the mitochondria to continue to generate the ATPs. Alright, the P50, you know the concept of P50? P50 is that partial pressure, that PO2 or that partial pressure at which the molecule or that particular chemical is 50% saturated. So P50 for hemoglobin is somewhere in the range of 25 to 28 mm of Hg. You can see in the graph, if this is 50% saturation on the vertical axis, 50%, 100%, 50% saturation, if you put a perpendicular on the hemoglobin curve, it will give you a value of 25 to 28 mm of Hg. But the P50 for myoglobin is 5 mm of Hg. Imagine 5 mm of Hg is a very low PO2. Until the PO2 has gone to 5 mm of Hg, myoglobin is holding half of its oxygen. So that further is a testimony to the fact that myoglobin is releasing its oxygen only when there is a very low PO2. I repeat once again, what is P50? That partial pressure of oxygen at which the hemoglobin or myoglobin are still 50% saturated. They are still holding half of their oxygen. That is the concept of P50, either for hemoglobin or for myoglobin. So P50 for myoglobin, just for the sake of your remembering, 5 mm of Hg. Okay, let's go through the remaining part. myoglobin takes up oxygen at low pressures, and much more readily it takes up the oxygen at low pressures. You can see here the graph is increasing, going up in the low pressures, and then at high pressures it is holding that oxygen. So at low pressures it will take up, it has got high affinity, 1 myoglobin is equal to 1 oxygen molecule, hence the percent saturation rises instantly. We saw that. Therefore it is rectangular hyperbola. One more point. What is the difference, cardinal difference between hemoglobin and myoglobin? Hemoglobin shows Bohr's effect. Myoglobin does not show Bohr's effect. Let's say, what is Bohr's effect? Increase carbon dioxide and hemoglobin readily liberates its oxygen. So muscle cell is contracting and generating carbon dioxide. Increase carbon dioxide means hemoglobin will liberate its oxygen readily. If this effect was also there with myoglobin, then it would interfere with myoglobin's ability to bind the oxygen. It will continue to liberate the oxygen. We want also myoglobin to hold the oxygen for some time. That would not have happened, and therefore remember, myoglobin does not have Bohr's effect like the hemoglobin. So even if muscle generates carbon dioxide and hence hemoglobin unloads its O2, it does not interfere with the capacity of myoglobin to take up the oxygen. Muscle is generating CO2, and so muscle needs extra oxygen because it is hypermetabolizing. Hemoglobin will give up its oxygen readily to the myoglobin, and myoglobin does not have that effect. Myoglobin will hold the oxygen because it does not have Bohr's effect. Even at the PO2 of 40 mm of Hg, myoglobin is almost 95% saturated, full saturation, and P50 for myoglobin is 5 mm of Hg. So these are the reasons why I said in the beginning that the myoglobin is an unsung hero and it's a part of very, very fascinating physiology.